All right, Jessica, you ready?
I am.
All right. So welcome everyone, especially all the Travis Chapins in the audience. We're excited to continue our new FAFP webinar series this afternoon and glad for the great turnout. We also want to thank all the members and sponsors for the ongoing support. So those are all on the screen right now. So just to start off with some housekeeping items, please keep your video and audio off throughout the duration of the program. We'll ask Dr. Chen to answer questions from the audience at the end of her presentation, and we have a team of volunteers who will be monitoring the chat box and facilitating your questions with Jessica. So please, if you have any questions, please enter those directly in the chat box. We'll also be recording the program and posting it on FAFP probably by the end of the week. That site has recently been revamped, so please check it out. As many of you know, we were supposed to have our annual educational conference in St. Augustine. That would have been back in May, but we had to cancel it due to COVID. And so instead, we've moved that programming online, and many of our speakers have agreed to participate. So today, our speaker is Dr. Jessica Chen. Dr. Chen is a microbiologist at the Centers of Disease Control and Prevention, supporting the National Antimicrobial Resistance Monitoring System. Her research focuses on understanding the molecular epidemiology and evolution of drug-resistant enteric pathogens. Prior to joining NARMS, Jessica was a postdoc at the University of British Columbia, where she collected comparative genomic research involving Listeria monocytogenes and Shiga toxic E. coli. Jessica received her PhD in animal science with an emphasis on microbial food safety from Texas Tech University in 2013. Jessica also holds a Master of Science in Animal Science from Colorado State University and a Bachelor of Science in Food Science from Cornell University. Jessica is Vice President of the Georgia Association for Food Protection, where she's also the chair of their program committee. Jessica's favorite quarantine activities involve playing board games, hiking Georgia's parks, and birding. And so with that, Jessica, I'll hand it over to you.
Well, thank you. Thank you so much for the opportunity to speak on this topic today. I've been really excited to share this material with you. So I'm gonna just give you guys an overview of antimicrobial resistance in foodborne pathogens in the era of next generation sequencing and kind of the experience that we've had and transitioning our surveillance systems over to WGS over the past several years. So a little bit about me, my academic background is in food and animal sciences, really specializing in microbial food safety with a kind of a specific emphasis on molecular biology. And so I came to CDC in late 2015 to work on antimicrobial resistance, where I kind of find myself in more of a bioinformatics analysis role. So it's been a role that has lended itself pretty well to teleworking, though it does get a little lonely. In my spare time, I took up birding last year, and the timing was impeccable because now it's good to spend a ton of time outdoors away from lots of people. So that's been one of my favorite quarantine activities. And I have two cats, Sigma and Mewtwo, that are pictured at the bottom, and they are keeping me company during these difficult times. So about who I work for. So I specifically, within CDC, I work for the NARMS program. So we're the National Antimicrobial Resistance Monitoring System. So NARMS is the tri-agency collaboration that conducts antimicrobial resistance surveillance in enteric pathogens. So this is done by, there's work across all three agencies. So there's a work that my group does at CDC that involves testing human isolates and looking at human clinical cases of infection. There's the work that's done at FDA that focuses on retail meat sampling, and then the focus at USDA that is really focused on what's going on in the food producing animals. So I'm going to give you the perspective from the human side today, so CDC's perspective, but FDA and USDA have both transitioned over to using this technology to study AR as well as do other things. They have experience and insight in this as well, but you're going to hear mostly from the CDC side today. So a bit of an overview about what I'm going to cover. I'll start out covering CDC's transition to whole genome sequencing and kind of tell you where we're at and how we got here and kind of give you an overview of the technology and how we're using it. I'll then give you a bit of background on the issue of antimicrobial resistance, the biology of antimicrobial resistance, and then launch into kind of NARMs and how we use WGS in our WGS story. And then I'll leave you guys with kind of the work that's just getting started, some of the future work that I think is the next frontier of AR genomics. So setting the stage on CDC's transition to whole genome sequencing. So this has largely been spearheaded by PulseNet, which is kind of the molecular epidemiology group that kind of connects the dots between these foodborne illness cases. So PulseNet, as you all probably well know, is a collaboration between CDC as well as our state and local public health partners. And it's been around since 1996. And when it first got started, they were using pulse field gel electrophoresis. People in the state public health lab would upload these patterns to the PulseNet national database. And then that national database would be mined to kind of look at where there are illness clusters, what isolates appear to be of the same subtype, and really kind of get at how and when outbreaks are happening. So in 2019, PulseNet officially switched over to using whole genome sequencing for its major pathogens. However, we are using WGS in a large way even prior to that. So in a lot of our outbreaks, we'd have a lot of sequences that we were then making kind of family trees of and looking at their relatedness and using that to help further our outbreak investigations, but we didn't really transition over that outbreak detection piece to whole genome sequencing until last year. So a bit about PFG, kind of where we came from, and I'll just go over this super fast because it's not something that's being used a ton anymore. It's still being used, but at least at CDC and within PulseNet, we've really fully switched over to WGS. So with pulse field gel electrophoresis, you get you extract the DNA and break up the DNA and the bacteria to release the DNA that are in these agarose plugs. They get cut up with these restriction enzymes and then run out on a gel. So you get this banding pattern, which you can call kind of a molecular fingerprint, so to speak. And so this is really all the information that we got when we would do PFG on an isolate. We would have a banding pattern that we could then compare with other isolates and be like, is this the same or not the same? Are these potentially the same organism or not? And with WGS, the wealth of the data that we get is just so much more. So we start again with a bacterial culture, just like we did with PFG. We extract the DNA. We chop that DNA up into pieces and then amplify it using PCR and then sequence those pieces. And then because they were chopped up into small fragments, the sequence data comes back to you in these short bits. And we process those, we call them reads, with powerful bioinformatic algorithms. And we can assemble those into kind of a reconstructed genome that represents like over 99% of the actual genome. So we have the entire genomic content that can then be mined further to gather more information. So often you'll see relatedness by WGS as illustrated by a tree, and here's kind of an example tree that kind of illustrates a typical tree that you would see as part of an outbreak investigation. So what you really want to focus on here is the genetic change. So that's like kind of the horizontal axes. The vertical spacing actually has no real meaning. It's just done to space things out. kind of some important terminology to kind of note when interpreting these things. So like you have clades, which are isolates that are descended from a common ancestor. So in this particular highlighted box, you have you know, these three isolates, A, B, and C, that are all descendant from this one node, which a node represents kind of a theorized ancestor. So that most recent ancestor for these three isolates. And then you have this kind of outgroup here, this isolate D. And it's actually not super straightforward to know kind of where to draw the boundaries of what's in versus out of an outbreak. And a lot of that has been kind of driven by the epidemiology over the years. So we have kind of informal thresholds that we use for the amount of genetic change that will kind of put something in or out of an outbreak. And we use those kind of as a guideline, because sometimes you'll see things that are different, and the epidemiology is informing that something really should be part of this outbreak. So different organisms can have different thresholds. Often in zoonotic outbreaks, we see a large amount more genetic diversity than you would see in, say, a point source food outbreak. And so those are kind of lessons that we've learned over the years, and things that we bring to our application and bring to kind of our whole genome sequencing investigations. So WGS driven outbreak investigations are not just done by WGS, but we really use that epidemiology as well, and it's really critical. So here's an example of kind of where that illustrates how much more information that whole genome sequencing is giving us than PFG, and how much better resolution this technology has. So on the right, I have a tree that's kind of a part of a tree from an outbreak associated with drug-resistant Campylobacter that was associated with pet store puppies. So not a foodborne outbreak, but it was an outbreak that we worked on extensively. You can see that these isolates are highly related in the tree. They're 98% similar. And so we have color-coded here kind of who the isolate belonged to. So the puppy emoji denotes isolates from puppies. The green emoji denotes a human clinical case that has an epi link to pet store puppies. And then the black-colored emoji denotes a human clinical case that was not epi-linked. So we didn't have any epi for that person. And then this code here on the right indicates the PFG pattern, and NP is not performed. So you can see that WGS is able to link all of these cases from humans, from puppies, whether or not they had epidemiologic information, even though they were put into different PFGE groups. So sometimes PFGE will split up things that don't need to be split up and actually appear really closely on the tree. And sometimes the opposite is true, where you'll get an outbreak where they're all the same PFG type, but you can actually see more discrimination in the whole genome sequencing tree. So I think that this is just like a really good case example of the types of information that we're getting through whole genome sequencing. And whole genome sequencing has replaced PFGE for cluster detection, so finding where those outbreaks are, but it's not only for outbreak cluster detection. We can find so much more out about what's going on in a given outbreak and what properties an individual strain has by digging into that whole genome sequencing data. So a lot of these wet lab tests that are done in labs have an in silico or a computational based analog that you can, like a software that you can run and kind of get the same result. So for example, serotyping. For Salmonella, there are like thousands of serotypes. It's really complicated. You have to keep around all these anti-sera. And you know, our partners at the University of Georgia actually developed this really great algorithm, and it's called Seq0, and it can serotype Salmonella based on its WGS sequence. And they just came out with a version 2 of that software and published that paper last year, and it's highly accurate. So things that we were previously getting through these wet lab tests, we can use those to get that same information through WGS. So, you know, subtyping, we can now build a tree and see the relatedness. So that kind of replaces PFT in that respect. And then things like MLST, we still may want to know something sequence type, and we can also infer that from the genetic data as well. Sometimes it's important to know what virulence genes are present. These used to be done by PCR, and now if you have a sequence, you can just mine the sequence for those targets. So as PulseNet was rolling out their plans to transition over to WGS, over in NARMS, we started to ask ourselves, well, can we get all of our antimicrobial resistance data from sequence? Can we harness all the isolates that PulseNet is sequencing and analyze those for resistance and be able to determine what antibiotics a bacteria would be resistant to? So now I'll just kind of switch gears a little bit and talk about resistance and kind of lay the foundation for some of the things that I'll be discussing. So kind of to give you a baseline idea of the scope and problem of antimicrobial resistance in this country. These numbers are from the CDC's AR threats report. So this report gets put out every few years and aims to quantify and kind of describe the state of antimicrobial resistance in the United States. As you can see, we have each year, due to antibiotic resistant bacteria and fungi, there are at least 2,800,000 cases of infection that linked to an AR pathogen, and that's associated with 35,000 deaths. And also, you've probably heard of Clostridium difficile. It's related to antibiotic use, and it's resistant to a lot of antibiotics, and that causes additional cases and death in the US. I'm going to focus mostly on enteric bacteria today. However, it's important to note that fungal pathogens can also develop resistance to antifungals, and these are a big problem as well. And there are teams at CDC that are working on this. So how do we quantify resistance? So there are many ways that you can get at this, but the gold standard technology that we use in the NARMS program routinely is this broth microdilution antimicrobial susceptibility testing. So on the right, you have a picture of one of our laboratorians, Christy Bennett, preparing a sample for AST testing. So she has a test tube here that has the bacteria and broth in it, and then a plate that has kind of these antibiotics at different concentrations. And so she dispenses those bacteria into the plate and then grows them in the presence of those antibiotics. And from the growth patterns, you can start to infer whether bacteria is resistant or susceptible to a given antibiotic. We can determine from this assay the minimum inhibitory concentration. So this is the lowest amount of drug that prevents growth of that microbe. So we have thresholds that say if it's resistant, if it grows in this drug concentration percentage or higher, it's resistant, whereas if it's below it, it's susceptible. But sometimes you have increases in the MIC over time that do not reach that resistant benchmark, and it's nice to have a handle on those trends to see, are those levels creeping up over time, because that's an important trend that we want to measure. There are additional tests such as e-tests and disk diffusion. I'm not going to get into these, but these can also be used to detect resistance, and they're great assays as well. It's just that the broth dilution MIC really gives us the information that we need, and it's our gold standard. So a little bit about our program at CDC and kind of what organisms we target and kind of how we go about doing that. So for surveillance, we look at non-typhoidal salmonella, shigella, E. coli, 0157. Those are kind of our major pathogens in addition to Campylobacter. We receive for those first three 1 in 20 clinical isolates that are submitted to state public health labs. So we get a 5% sampling of all salmonella, all shigella, and all E. coli 0157. In addition to that, we get typhoidal salmonella and Vibrio non-cholera submitted, like all of the clinical isolates corresponding to these pathogens sent to us. And then our Campylobacter sampling actually varies by state. Not all states isolate Campylobacter, and so we're dependent on the ones that do to forward us some of their Campylobacter for AST testing. In addition to kind of our routine NARM surveillance, we look at outbreak isolates as well. So we get 3 representative isolates from multi-state outbreaks and analyze those as well. And then notably, this is just phenotypic surveillance. I'm not even getting into the process for WGS. So a little bit on how bacteria become resistant, because they don't just kind of magically become resistant. This is really driven by their biology. There are many ways that bacteria can do this. And this is kind of a very simplified version because it's super easy to get in the weeds, and there are tons of different mechanisms by which bacteria can acquire resistance, and we're always learning about new and novel mechanisms, and it's a very big field. So kind of very broadly, bacteria can do things like pump antibiotics out of the cell. They can modify components of say like a receptor within the cell so that it no longer binds with the drug and the drug can no longer act effectively. Bacteria can restrict access by changing the way that antibiotics can enter the cell. They can actually destroy the antibiotics with enzymes that they acquire. And they can develop new cell processes that avoid using that antibiotics target. So those are some of the broad ways that the bacteria will become resistant to antibiotics. And then kind of how this actually occurs, there's like a mechanistic factor where, how the bacteria actually respond, but how do they acquire those mechanisms? This kind of falls into 2 broad categories. So you have acquired resistance on the left-hand side, which is driven by resistance genes. And so these are often things that are mobilizable and that can move around from bacteria to bacteria through a process called like horizontal gene transfer. So these genes are often located on mobile elements. Most famously, at least in our organisms, this is commonly plasmids, so these circular pieces of DNA that can move from cell to cell, which I have illustrated here. So you have this blue bacteria that has a plasmid, and that plasmid can be transferred to the red bacteria. Now they both have that plasmid, which plasmids may not always contain resistance genes, but they can, and this is a big way that resistance genes move around. Also, this often occurs in response to being exposed to an antibiotic. A bacteria can develop these single nucleotide polymorphisms. These often occur in the chromosome in sort of in a housekeeping gene that's like an essential part of the core genome. And these mutations can be very stable and inherited by all the offspring of that particular bacteria. So now I'm going to move, now that I've kind of set the stage for this, I'm going to move into how we track antimicrobial resistance using whole genome sequencing and how we got to this point. So a few years ago that we asked ourselves, this is a royal we, this is actually like pre my time at CDC. We asked ourselves, can we predict antimicrobial resistance phenotype using this genetic sequence data? And so I kind of highlighted a number of the NARMS publications really getting at this. And, the answer was really a resounding yes. Yes, we can do this, and we can do it with high accuracy. And the reason we can do this is basically due to that genetic basis of AR. And I was kind of getting at this a little bit earlier. It's all embedded into the bacteria's genetic code. So as long as we know what we're looking for, we can very accurately predict resistance. So for example, these colored boxes here represent resistance genes. This big circle here is your chromosome. The smaller circle is the plasmid, which the bacteria may or may not have. And then you have these mutations that I mentioned that are occurring chromosomally occurring housekeeping genes. So this is kind of a normal gene without the mutation, and the star denotes the mutation. And by comparing these patterns to our genomes to a database that kind of defines what are resistance genes and what are resistance mutations, we can kind of put those things together to get a complete picture of antimicrobial resistance. So we refer to these mutations in genes collectively as resistance determinants. So these are basically genetic elements within the genome that confer resistance to a particular antibiotic. So kind of a high-level overview of how we do this. In PulseNet and in NARMS, we rely heavily on Illumina sequencing technology, though there are other sequencing technologies out there that are very useful. We also use MinION as well for more researchy projects, but most of our day-to-day sequencing is done with the Illumina instrument. We run our sequences through some QC checks to make sure that this is the right organism, that, when someone submits to a salmonella, that it's actually a salmonella and the serotype is what we would expect. We assemble those sequences into kind of a reconstructed genome, which we call an assembly, and then we find those resistance determinants. So those genes that confer resistance, or those mutations that we know also confer resistance. So once we get kind of that constellation of resistance determinants that we have, when they're present, we know what determinants cause resistance to what antibiotics. So this was done through a very painstaking process of going through kind of a lot of our validation data, but also the literature, and kind of looking at kind of what phenotypes and are we going to expect when we see this gene is present or this mutation is present. So we have this gigantic drug key, like in our database alone, there's like 3,000 resistance genes. So we know that when we see this particular gene, we expect this, this antibiotic resistance profile. And that's, like I mentioned, driven mostly by our validation and our in-house experience, kind of day-to-day seeing this confers resistance to that. But there are some determinants that we're never going to see in enteric pathogen, or we might not see for a long time. And until we receive an isolate with that gene, we actually don't know, and it's our best guess based on the information that we can find in literature. So this is an example of kind of how we do this. So this is Salmonella Newport from our routine surveillance. It picked up this BLA-CMY gene, which we know confers resistance to these drugs here. It picked up these two genes here, which are aminoglycoside resistance genes, and they confer resistance to these drugs, and so on. So from just this profile of resistance genes and mutations, we can then say these are the resistance, the phenotypic resistance that we would anticipate for this isolate. And we typically refer to this as predicted resistance, because it's not actual phenotypic resistance, but something we've inferred from the genome. So hopefully I've kind of convinced you like that whole genome sequencing really is antibiotic resistance prediction. It's a really consolidated workflow. It's actually, the turnaround time for this is much quicker than getting these isolates into our lab and performing phenotypic testing. So there's a decreased cost and effort associated with this, and it actually allows for more real-time testing, and I have some slides that will illustrate this in a little bit. We get a lot greater detail, and so this image on the right, is showing what we would get with phenotypic testing. So this is our 5% sampling of, say, Salmonella that our state public health partners sent to us, and we only have phenotypes. So this is resistant to a drug called ampicillin. But on the right, you have a picture of kind of what we get in the era of whole genome sequencing. So we have not only access to the 5% of isolates that we receive in our lab, but we have access to the sequence for everything that's been sequenced at state public health labs in the PulseNet network. So we can analyze all those and say, this gigantic pie here is actually all the ampicillin-resistant isolates, and these are the individual genes driving resistance in each case. So yeah, so that's kind of some of the advantages, and I'll get into in a little bit kind of how we use this information and why it's useful, particularly in an outbreak setting, but also for surveillance. So I think it's important to note that NARMS will continue to be doing phenotypic testing for some time. Really, PulseNet's transition to whole genome sequencing has increased vastly the amount of genetic data that we have, and that allows us to identify really rare resistance occurrences, which we didn't always have the power to do before when you're only getting 5% of the clinical isolates for a given pathogen. And it also increases our ability to detect changing trends. But really keeping that phenotypic data is really helpful to allow us to investigate genotype and phenotype discrepancies. As I mentioned, we are heavily reliant on our database to determine what is a particular resistance gene versus what's just, another gene causing another function in that bacteria. So when we see something where the phenotype says it's resistant, but our genotype doesn't have a resistance element for that, we start to investigate and to dig deeper and try to figure out why this isn't lining up. And so an example of this work is last year, we identified a new trimethoprim resistance gene, DFRA. This is now in our database, making our whole process more accurate. It's already highly accurate, but really keeping around that phenotypic testing allows us to continuously refine the prediction that we're doing. And this next slide really highlights, I think, in a single image, like how much this has improved our ability to respond to public health challenges. So I kind of mentioned earlier, there was a Campylobacter jejuni outbreak linked to puppies. It was actually featured on the Colbert Show. I think he called us like the CDC cheer stompers or something like that. But yeah, everybody was really sad to see this outbreak linked back to pet store puppies. But really, like, NARMS was able to find out a lot about this pathogen before we even got isolates in the door. So we have this outbreak being identified on August 21. 2 days later, we have sequenced isolates in our database ready for screening. 5 days later, we actually screened the isolates. I honestly can't tell you why it even took us that long. It was probably my fault. But so within a week, we have results that tell us basically what this pathogen is going to be resistant to. On September 8th, so then the following month, we have the isolates arriving at CDC for AST, and then we have the AST results available. So we have data on this 1 month earlier, which in the context of an outbreak is super key. And in the context of this outbreak, the pathogen was resistant to nearly every antibiotic on our panel. So including like all of the treatment options for CAMPy. So there was this need to start testing drugs pretty quickly to find treatment options, because normally campylobacteriosis is self-limiting, but not always. People can get severely ill and sometimes do need antibiotics. And in this case, we were able to say, we have a limited amount of data to say that in this case, meropenem works. Whereas if we hadn't had that month lead time, we would not have been able to respond to this nearly as quickly, and we would not have had that information to give to clinicians to help drive the treatment of patients that are coming down with infections related to this outbreak. So a little more on kind of how we use this data. So like I said, we have this resistance determinants pattern. We also actually look at plasmid subtype. That can be a useful indicator too. And we infer that predicted resistance pattern. So where these determinants come into play, they're really useful for subtyping. They kind of are much more, offer a much more granular view of what's going on resistance-wise. You don't just know that something is resistant to a particular drug, you know exactly what's driving it. You can get out a little bit of source attribution too. We've done some studies in the past where we associate certain plasmid types and certain genotypes with, say, a cattle source or a chicken source or a puppy source, for instance. So these, knowing these specific determinants is really useful for that. And then also from the genetic data, we get information on the context, the plasmid and mobile elements. So we know if the resistance that we're seeing is chromosomal, it's not going to move around as freely as if it's on a plasmid. So it's important for us to know that as well. With the predicted resistance pattern, it's nice because we can use that to link back to our historical data. Have we seen this resistance pattern before? Because our NARMS data goes back 20 years. So it's useful to have that predicted resistance so then we can query our database and be like, what matches this? Because prior to, say, 2015, we don't have a ton of sequencing data. It provides some weak subtyping, some source attribution, but not nearly to the level that we see when we're looking at the actual determinants that are driving the resistance. And then it also gets at that clinical importance. So we know, what this is to be anticipated to be resistant to. We can share that information publicly about kind of what treatment options may or may not be on the table based on the genetic profile. So this leads nicely into kind of where our data goes. So our data goes onto our public-facing website. It's called NARMS Now. And I borrowed the slide from my boss, and the note says, let's be honest, used to be more like NARMS eventually, but it's actually NARMS now, which I think is pretty funny. It took us a while to kind of get to that point and build out all the pipelines and stuff to be able to pushing that data up. And now it happens nightly. So you can see our up-to-date information on NARMS Now. It's a really cool dashboard. I encourage you to check it out. You can filter by pathogen. You can look at the resistance to a particular drug over time. So our approved and cleared data is going into this NARMS Now database. We also put this information into outbreak web postings. So you may have seen some of these postings that CDC puts out when they're investigating multi-state outbreaks. Like this top one is romaine. We had an outbreak linked to raw chicken, raw turkey. And if you dig into these, it'll say stuff like, so many number of isolates are predicted to be resistant to, X antibiotics. And that is NARMS data that's being kind of put into these web postings to really inform clinicians about what resistance is potentially part of this outbreak. And then once we get that phenotypic data confirming the result, that information goes in as well. We've been looking at, there are a number of outbreak illness clusters that have been associated with resistance. So I mentioned the puppies a couple times. There has been a Salmonella redding that has resistance. It's been circulating, that's been associated with turkey. There are multiple outbreaks going on with backyard poultry, they tend to peak in the summer. Multiple serotypes in some of them. We see more frequently resistance. Dairy calves, guinea pigs, there's a lot of stuff in here that's not food, but they're animals that we come in contact with. Chicken salad, romaine's a big one that's on people's mind. That had resistance as well. And then we have also been monitoring extensively drug-resistant typhoid and returning travelers. So this is kind of just a sampling of some of the things that we see and respond to. So whole genome sequencing really allows us to better understand the ecology and spread of resistant organisms in a way that previous technologies didn't allow us to do. So I've illustrated on the right here, this is a time tree of multi-drug resistant Salmonella infantis. This is similar to the trees that you've seen previously, but the scale is actually due in time. This is not a type of analysis that we do routinely, but we do in kind of special cases when we want to get an idea of kind of how something is spreading over time, something that we've seen multiple years in a row, and we want to have a better sense of kind of what's driving this. We'll put it into one of these, it's called a molecular clock, to understand what's going on over time. So this is a strain that we initially saw in returning travelers. So we compared it to this publicly available genomes that we get from a public database. And we were able to see kind of that this strain originated in South America, that you can see some these cases that are highlighted in black here are isolates from the US. Things in blue are from Peru. And there were a lot of Peru sequences in the database. And then you can see kind of there are these like little sporadic introductions that are only a few cases, and you have this big clade up top. And this big clade up top corresponds with the emergence of this strain in chickens. So we started seeing a rise in illness among non-travelers. So that kind of the epi really shifted around 2014, 2015, where we started to see this particular organism more in people who did not travel. And then our partners at FSIS started detecting this strain in chicken samples as well. So kind of putting these things together and seeing how things changed over time was really helpful to us in this investigation. And this strain continues to be associated with ongoing illness. And this presents a big challenge for us because a lot of the times we think of outbreaks as these point sourcey things where we can rush in and you know, identify the food vehicle and recall it. But when it's one of these things that kind of lingers over time, this particular strain seems to be associated with, you know, multiple plants, multiple producers. It's not a single source. It seems to be something that's really distributed far throughout the production chain. So, you know, some of the ongoing conversations that we're having at CDC in the area of whole genome sequencing is, how do we really track, trace, and control these things that seem to be recurring? E. coli 0157H7 in romaine lettuce is an example. It seems to be something that keeps cropping up, you know, and it's like, how do we respond to this? How do we adequately track this? Because a lot of our outbreak framework is really formed around responding to these point source outbreaks. So these are kind of some of the questions that we now have, now that we've kind of peeled back the curtain and start looking at these things in great detail and have the resolution to really track these things. So what's next for AR genomics? This is going to be my last piece, and then I can take some questions. So as I mentioned, in many years, we've been dealing with a lot of big systemic outbreaks that were multi-drug resistant and keep kind of cropping up over time. And so these are some publications with some of these different multi-year, multi-drug, multi-state outbreaks. And we find that this resistance is often mediated by plasmids. And sometimes these plasmids are common carriers of resistance, and other times they are new plasmid types that have never been seen before in the serotype. So there's kind of a lot, these outbreaks tend to catch us by surprise. And kind of the reason for that is kind of the way that we've been conducting surveillance. And so, you know, as I mentioned, we have initially started out looking at just the phenotype, and we can map these trends over time, and we could easily see how our resistance to certain drugs was rising and falling over each year. And then we added that next layer, that whole genome sequencing layer. So this is the era that we're in now. We have really fine-grained resolution that allows us to kind of pinpoint what resistance is being driven by. But what we really don't have an idea about is the ecology, right? So that layer underneath, what's driving all of this? How are these plasmids distributed in sources beyond humans, beyond food animals, beyond food? Where are these things coming from? And part of the challenges part of the big challenges, we don't really have a great way to classify plasmids. Like, we have great ways to classify bacteria. We can put them in a tree. We can say these things are related. We have a group that's developing these new allele codes that are like a zip code that'll say, you know, these two isolates are the same, right? So we have really good ways of looking at kind of the isolate, but we don't have great ways of saying this is the same plasmid. And it's very, right now, that process is very artisanal and very time consuming. So to get at this challenge, we've actually teamed up with a group led by Fernando de la Cruz. He's a well-known plasmid biologist, to kind of validate and implement a better way of classifying plasmid. And it takes into account the whole plasmid and not just single genes, not just the resistance genes, and it clusters them based on average nucleotide identity. And so this one allows us to create a plasmid taxonomic tree like you see on the left. So you can start relating plasmids in the way that we've put bacterial isolates into a tree. It allows us to uncover these patterns of plasmid movement. So this chord diagram on the right is showing the distribution of different plasmids and different organisms and kind of how it's connecting kind of similar plasmids across those genera. So it's really illustrating how these plasmids move across species boundaries. And so it'll be really great for us to start knowing how these resistance plasmids move into foodborne bacteria, because right now we're not, CDC isn't sampling a lot of these things because they're not foodborne pathogens, right? Or they're not pathogens. So there's so much to the ecology of resistance that we're just starting to understand. And so this work is really just getting started, but there's a lot of potential for this tool to really improve our understanding of this ecology. And we hope that it will lead us towards that kind of a risk metric that can start be using in a public health setting. How important is this plasmid? That is it really capable of moving about, and how concerned should we be about it disseminating throughout foodborne bacteria? Kind of the next major challenge is metagenomics. And so this is something that we are working on with another team within our branch. So we have an entire metagenomics team that's doing a lot of super great work. And the reason that metagenomics is really important is we actually are starting to get fewer and fewer isolates sent to us. So all the work that our surveillance system is doing now is really based on sequences of single isolates. And this diagram here illustrates kind of how an isolate gets to us, right? So you have a sick individual that may go to their doctor. They'll provide them a stool sample. This is a disease stool, so it's sad. It goes to the clinical lab where it's isolated, and that isolate is forwarded to the public health lab where molecular subtyping may be done. And then we analyze that sequence, both PulseNet and NARMS, to know whether it belongs in an outbreak. We look at it for its resistance profile. But in this current age, we're seeing less isolates being forwarded to us because of these culture-independent diagnostic tests. So these are things that are tests that are actually available at hospital lab that are a PCR-based test or some other rapid technology that will give you an answer in minutes, but it actually doesn't tell you what the isolate is. So you put the sample in, it tells you what bacteria is present, but it actually doesn't tell you, it doesn't give you an isolate out on the end. It bypasses the whole isolation step. So you can see that, the sample goes to the clinical lab where it gets run on a CIDT. That ID information goes back to the doctor. Your stool may get sent to the public health lab, but it's not necessarily isolated, and then it leaves, who knows, for surveillance. So we need to find a way to continue to be able to get this kind of information. And so this is my last slide, and we are using some of these technologies to look at antimicrobial resistance direct from stool. So this first panel here, is that Amplicon panel. So this is basically, this instrument does basically a lot of PCRs to amplify out very specific targets that we're interested in. So in this case, resistance genes. So it'll amplify just those resistance genes in this magic box that's called a fluidime Juno, and those get sequenced, and we can then infer from that what antibiotic resistance determinants are present in the stool. A challenge of this is that there can be lots of bacteria in your stool. They might not just be pathogenic bacteria. They can include bacteria that aren't disease causing, but they'll have resistance genes as well. And we don't have a good way of detangling what is pathogenic versus what's not, right? So that's where this high-C technology comes into play. It's just kind of coming into use. It is able to cross-link the DNA for a given cell. So you start getting DNA that's all grouped together, and that you sequence that and are able to tell what organism that read came from, because usually you're sequencing very small fragments of the DNA. Sometimes it's hard to tell, particularly if it's a plasmid, what organism it belongs to, right? So with this technology, we can start to say, these are the resistance genes that are present, and this is the organism that it's in. And then CDC is also doing a lot of finding some bioinformatics solutions to use with both of these technologies, and there's a lot of effort going into that. So these two kind of research pathways are ways that we're trying to get at that problem of the fact that we're not going to see as many isolates in the future, but we still really have a need to detect resistance. So that's kind of a big flyover view of the work that we're doing in this space. If you're interested in learning more, NARMS, our tri-agency collaboration, is holding a virtual public meeting October 13th through the 14th. If you're interested, like, let me know. I can give you more information on how to get registered for that. But there'll be a lot of different stuff. We'll talk more about resistance plasmids. We'll talk more about metagenomics. It's really kind of the work that NARMS is doing now, and how we're going to keep improving in the future. And with that, thanks for your time today. I appreciate you coming to listen to this talk on AR. And I'm happy to take any questions at this point. And if you think of anything afterwards, like, feel free to email me. My email's on the screen. So thanks, guys.
Thank you so much. There are a few questions here.
Sure.
The first one, are there outbreaks where the isolates have different AR profiles?
Yes, so this can happen. Sometimes it's kind of, so sometimes they're more minor differences, but sometimes they're more drastic. So sometimes we'll see isolates that have lost a whole plasmid at some point in the way, or kind of never acquired that plasmid. So you'll see things that have like maybe a resistance to like 5 drugs, but then some that don't because they've lost that plasmid or have never gotten it. And sometimes we'll see things like there was the outbreak associated with dairy calves. There were some isolates that had fluorphenicol resistance, some that didn't, but all of them had the resistance gene. But when we dug in deeper, we found that this transposon had jumped in and inactivated the resistance gene in a sub portion of the isolates.
The next question, sorry, was, does NARMS only look for AMR in pathogens? Any attempt to look at non-pathogen hosts or reservoirs of AMR?
Yeah, so CDC really looks at, is really focused on kind of the human clinical isolate, clinical angle. However, some of the other groups of NARMS are sampling some kind of indicator organisms and other pathogens, so in other settings. So for example, like FDA is looking at generic E. coli, and we just focus on 0157 because that's clinically what's important to us. We also do screen other shigotoxigenic E. coli and look at those, but we don't bring them into our phenotypic sampling. There's actually movement towards building an environmental arm of NARMS. So this is going to be done via collaboration with EPA and CDC's waterborne group. And that is going to be really interesting because it's going to be sampling waterways for resistance as well, kind of bringing in a whole nother avenue. And we're always talking about how to bring in new kind of sources of samples. So I think FDA is looking at kind of what other food products they can be sampling to look at AR, because we kind of focus on some kind of major commodities, but I mean, there are a lot of different commodities that can be playing into this that are kind of gaps in our system that we're looking at addressing.
That's great, thank you. That's all the questions for right now, and I'll hand it back over to Jason.
All right, so just thanks for the presentation. I'd just like to make everyone aware that there are 2 more scheduled talks, September 2nd, September 16th, and I believe we're also looking at having a kind of an AEC business meeting in early October. So please be aware of that for an opportunity to provide some input in the direction of the organization. I'd also like to make sure that you check out our new FAFP website so that if you're not a member, this is a place where you can join It's actually a pretty low cost. We'll also be posting all of the presentations that we've had so far. Those are all going to be available here at the website. So with that, thank you for joining us. Hopefully we'll see you in a couple weeks.